Okay, welcome back to the continuation of the cardiovascular physiology lecture. And last we left off, we talked a little bit about the depolarization and repolarization and something of the upward and the downward deflection of the stylus and what that means. So now we're going to look at it from an anatomical standpoint so you can understand that a little bit better. Let's just start with the layers of the heart. So if we were to look at the heart over here in general, and you just see we're to take this section right here. This whole section of the heart, which is the end of the heart, inner towards the outer side of the heart, and then we zoom in on this picture right here. Okay, we have different layers. We have right here, we have the endocardium, endo meaning most inner layer, right? The myocardium is the muscular layer, and then we have the epicardium right here on the outside. So let's talk about these three different layers. And then there's that fibrous capsule on the outside. But when we talk about layers of the heart, we're going to talk about the endo, myo, and epicardium. Okay, so the endocardial cells, it goes from endocardial cells to myocardial cells to epicardial cells. We go from inner to muscular to outer. So when this wall depolarizes, it depolarizes from endo, myo to epi. So it goes in this direction. It goes from endocardium to myocardium to epicardium. So if the electrode is right over that tissue, right over here, over that tissue, depolarization goes from endo to myo to epi towards the electrode. So it's going towards the electrode. So it's going to give a positive deflection of the stylus. Now, repolarization, remember, does not occur in the same order. What's going to repolarize first? We're going to go in the opposite direction, where the epicardial cells are going to repolarize first, then the myocardial cells, then the endocardial cells. So depolarization, we have this electrical signal going through the heart, right, into the Purkinje fibers. It's going to go this way. But then when we repolarize, we just depolarize. So where do we have to start? We have to start on the outer layer and then go back in to repolarize. So this is why the depolarization and repolarization is opposite with regards to the deflection of the stylus. So when the ventricles are repolarizing, we're going to start over here. We're going to repolarize this way because we just depolarized this way. So now we have to start here to repolarize all these cells. Now what's happening? Depolarization was going towards the positive electrode, so we're going to have an upward deflection. So now if we're going to repolarize and we're going away from that positive electrode, we're also going to have an upward positive deflection of the stylus, right? This is why the R and the T waves are both positive. So this is all related to ionic movement. So it's not just random that the waves are positive and negative. It's all based off of depolarizing and repolarizing with the amount and the layers of the cells that it goes through. So if we go again, we're going to depolarize, right? We send this electrical signals down through the the SA node, the AB node, the bundle of His, then we go down to the bundle branches and the Purkinje fibers and all that, and we send that signal through to depolarize these cells, right? We're going to go from endo to myo to epi, and if we had a positive electrode over here, we're going towards that positive electrode. So we're going to have an upward deflection of the stylus. Now when we repolarize, we have to repolarize in the opposite direction because we just depolarized here, so we need to repolarize back this way. And if we're going away from that positive electrode, we're also going to have a positive deflection because we are repolarizing in the opposite way and it's going away from the positive electrode. Okay, so when we start talking about leads and vectors, a full ECG or electrocardiogram has 12 leads. We have 6 limb leads and then 6 chest or what's also called precordial leads. Okay, so up here we have our limb leads, and then down here we have our chest leads. Okay, so when we talk about 3 of the limb leads, which are bipolar, 1 electrode is positive and 1 electrode is negative. We are looking for the electrical difference between them, and this is all related to ionic movement. So we have, right here, we have lead 1, lead 2, and lead 3. All right, this should be 1. Okay, so with lead 1, we're looking at the difference between the right arm and the left arm, and then we're going to designate that as negative and that is positive. The right arm and the left leg, which is lead 2, and then the left arm and the left leg would be lead 3, right? And now we're looking at the potential difference between these these different electrodes, and in there is going to be all related to ionic movement, and that's going to determine if we're going to get a positive or negative deflection of the stylus. So, all right, so now if we go to the next unipolar limb leads, they're called augmented limb leads. We have AVF, AVR, and AVL. Here we're going to designate one of the leads to be positive and take the average of all the others and make it a negative. That negative will be put in the center right here. It is considered a unipolar lead. We are looking at one terminal in the center, and this is called the central terminal of Wilson. We use this terminal as a reference point and compare everything else to it. So we place these augmented leads 30 degrees in between 1, 2, and 3. So if we had lead 1, lead 2, lead 3, we're going to put these augmented leads 30 degrees in between those. The positive electrodes are located on the left arm, which is the AVL, the right arm AVR, and the left leg AVF. These are the same electrodes used for 1, 2, and 3. Okay, so electrode placement. So with these limb leads, we're going to put 4 electrodes on an individual to get 6 pictures. Here, 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 and this is going to be considered the ground, right above the iliac crest over there. Okay, we get 6 pictures of electrical phenomenon as it moves through the myocardium, and we look at these 6 pictures in a frontal plane, right? So the lead placement, the shoulders and the hips while we exercise. So sometimes you may see these leads may go on someone's ankles, right? But we need to shift those and we not to have them on the wrists or the ankles. We need to have them right below the clavicles and right above the hips, because we want to be able to exercise while recording different data. Okay, so we are looking at these, again, we are looking at this heart, the heart in a frontal plane. So if we were to cut the heart in half, but from a frontal or coronal perspective, it makes front and posterior sections. So it's looking at the heart in that type of a plane. Okay, so when we're talking about leads 1, 2, and 3, lead 1, we're looking at the electrical potential difference in between left arm minus the right arm. Lead 2 is left leg minus the right arm, and lead 3 is the left leg minus the left arm. So if we were to add the voltage in lead 1 to the voltage in lead 3, we will get the voltage in lead 2. So lead 1 plus lead 3 equals lead 2. The right leg is used as a ground, and that's just for recording purposes on the ECG. Okay, so now we will talk about the chest leads. Okay, so these are considered 6 unipolar leads, and lead placement all goes on the chest. So here, with the precordial of the chest leads, we are looking at the heart in a horizontal or transverse plane. So if we were to slice it in half in a transverse section and going into top and bottom halves, lead placement goes on the chest here. So it goes front to back 6 different times. So when we're looking down at the heart, what's occurring is that signal of depolarization and repolarization. When we cut the heart in half in a transverse plane, it's going back and forth 6 times, front to back and forth 6 times, and giving us a reading. Okay, so unipolar. The entire body is used as a ground, and we compare that charge to the positive charge of the electrode. Okay, so I'll say that again. They are considered unipolar, and the entire body is used as a ground, and we compare that charge to the positive charge of the electrode. Okay, so the placement for the chest leads are the V1 is in the 4th intercostal space on the right of the sternum. V2 is in the intercostal space on the left side of the sternum, so right opposite 1 is 2 on the left side. We're going to hop to V4 quickly, because V4 is in the 5th intercostal space in the midclavicular line, right? So if we line up here, midclavicular line is going to be 4. So it's easy to do 1, 2 placement, and then 4. It's in that 5th intercostal space. And then in V3 just gets placed in between V2 and V4. So when you are eventually placing these on each other and individuals, 1 is in the 4th intercostal space here, and 2 is in that 4th intercostal space, and 4 is in that midclavicular line in that 5th intercostal space, and then you would just put lead 3 right in between 2 and 4. V5 is put on the anterior axillary line in the 5th intercostal space, so kind of right across from 4, but we follow the ribs around and we go into that 5th intercostal space, but it's right in line with the armpit here, that anterior axillary line. And then the mid-axillary line is 6. Okay, so this still in the 5th intercostal space, we're following the space around, but instead now we're in that mid-axillary line, which is actually closer to the armpit, and the anterior axillary line is a little bit over, right? So a little bit over from the midclavicular line. That is the chest lead precordial placement. Okay. All right, so next we're going to talk about vectors. The electrodes are placed based on how they represent vectors. So a vector can be used to represent cardiac electrical activity at any one point in time. They show two things, direction and magnitude. So the direction and propagation of cardiac electrical activity is represented by the orientation of a vector. The ECG recorder interprets these vectors and turns them into waveforms. So which is what we see on the ECG recording. So really those waveforms are just vectors within the heart. All these vectors move from an endocardial surface to the epicardial surface. So in the final video of this the cardiovascular physiology lecture, the next video, we will sum up what the mean QRS vector is, what that means, as well as the 3 major vectors that are pictured within the heart, and then as well as we'll just kind of do some summary statements about vectors. Okay,